Everyone knows the story of the Lion King and the story of Simba. Who can forget his dignified roar on top of Pride Rock? But have you ever wondered how Simba got his powerful roar? Well, let me tell you. It all starts with protein synthesis, the process where an organism generates new proteins. Simba's roar gene is made by the central dogma, DNA to RNA to protein. This process allows Simba to make a roar gene, which gets translated into a protein, making his roar loud. The step DNA to RNA is also called transcription. The process of making an RNA copy from a DNA template is the process similar to replication. Transcription is completed in 3 steps. The first is initiation. During initiation, RNA binds to a location on the DNA called a promoter. The promoter signals the beginning of a gene. The promoter is therefore the start codon and is where the RNA polymerase attaches. The end of initiation is where RNA polymerase unzips the DNA. The next step in transcription is elongation. Elongation is where the RNA polymerase makes a complement for one of the DNA strands. During this step, tRNA searches for a complement codon on the antisense strand to pair up with. A codon is a set of 3 base pairs. It codes for a specific amino acid. Since Simba is a living organism, he has eukaryotic cells, which means the mRNA transcript must be edited. When you edit, you have to take out all the introns. You have to do this because introns don't code for anything. Once you take out all the introns, you must splice the exons together. Once they are all together, a cap will attach to the front of the exons, and a poly A tail will attach to the back of the exons. This gene is now mature because all the introns are gone. Now we move to the process of translation. This is the process of where you decode mRNA into proteins. This process has 3 stages. The beginning stage is initiation. In initiation, translation begins when a ribosome attaches to an mRNA molecule in the cytoplasm. So translation begins at the start codon, AUG. Every time tRNA pairs up with its complement, an amino acid attaches to another amino acid in the P site of a ribosome, making an amino acid chain. To complete the amino acid chain, the ribosome works like an assembly line worker who attaches one part to another. The ribosome helps form a peptide bond between the first and second amino acids in the A site of the ribosome, while at the same time, in the P site, the bond holding the first tRNA molecule to its amino acid is broken and exits the ribosome. This process is called elongation and is continued until the 3rd stage of transcription, termination. Termination is when the process of translation is stopped. The polypeptide chain continues to grow until the ribosome reaches stop codon. All the DNA stop codons are UAA, UGA, and UAG, but the sequences will only use one. Then it releases both the newly formed polypeptide and the mRNA molecule, completing the process of translation. Now imagine if Simba's roar still sounded like this when he was full grown. We assume his genetic code had some point mutation along the way. A point mutation is a gene mutation that involves changes in one or few nucleotides. This could affect Simba's roaring gene because it affects a single point in the DNA sequence. A point mutation includes substitutions, insertions, and deletions. These kind of point mutations can change Simba's loud roar into a little roar, because by changing one specific gene, you can change the entire reading frame.